Hey guys, Kenny here for another round of new material. So today we're going to go into a little bit of a different direction. We're still talking about inheritance. We're still relating this to dominance and recessiveness. And while last class we were really focusing on traditional inheritance, where you had a dominant trait that would overpower the recessive trait and be able to hide it, we're going to look at some examples of inheritance that don't follow that traditional pattern or those statistical inheritance predictions. So when we go ahead and take a look at this, let's go ahead and start with the essential questions. It's too far. Number one, how are codominance and incomplete dominance different and the same with respect to Mendelian inheritance? Now, the Mendelian inheritance is what we talked about before in that traditional inheritance pattern. Number two, how are polygenic traits, you know that prefix poly, different from monogenic traits? You should know that prefix mono. Think monomer, polymer. And number three, why do you get different ratios when dealing with lethal alleles? Okay, so let's go ahead and go over some of our key vocabulary again just to make sure that we're all on the same page. Let's start out with genotype and phenotype. Remember that the genotype of an organism is the actual genes themselves that go ahead and carry the instructions. Okay, so when we start looking at a particular allele in the example that we're given here, you have the genotype big P, big P. Okay, those are two dominant alleles for P, which gives you purple. Okay. Likewise, you have big P, which is heterozygous, and little b, which is homozygous recessive, that gives you the white. Now, the color purple or the color white, these are what we call phenotypes. These are the actual observable features or traits. Now, there can be more than one genotype for a given phenotype. For example, on the right, you have the homozygous dominant, big P, and the heterozygous big P, little p, both of which give you the color purple. Okay, this is because the dominant trait is able to mask or cover up the recessive trait. By convention, the dominant alleles are labeled with a capital letter and the recessive ones with a lowercase letter. A heterozygote then is going to be identified as a carrier, and this is an important vocabulary word to make sure that you understand. A carrier is someone who has the recessive allele but doesn't show it. And oftentimes, carriers are unaware that they carry these recessive alleles until they go on to have children. Last class, we also went ahead and focused on understanding how to use a Punnett square, and we gave you this example you see here on the right. We're crossing two heterozygous parents, and we separate out their alleles, big P, big P, little p, and our Punnett square gives us the option of 3-to-1 ratio between purple, okay, the dominant trait, and 1 for white, the recessive trait. Okay, this 3-to-1 ratio ends up being really common in Mendelian genetics because the dominant masks, and in the intermediate version or the heterozygote shows the dominant trait. So let's do a quick summary of what we've learned from Mendel at this point before we start making things complicated and introducing codominance, incomplete dominance, and other forms of inheritance. These are Mendel's principles, and I'm going to go ahead and break them down into some key points to hopefully kind of summarize or generalize the ideas that we covered before. Number one, the inheritance of biological characteristics is determined by individual units, okay, what Mendel called factors, but we now call genes. In organisms that reproduce sexually, genes are passed on from parents to offspring. And what we typically end up looking at are the alleles. Remember, alleles are versions of a particular gene. Number two, in cases in which two or more forms of a gene of a trait exist, some forms of the gene may be dominant and others recessive. This is often referred to as the law of dominance, okay, as the dominant trait will mask or cover up the recessive one. Number three, in most sexually reproducing organisms, each adult has two copies of each gene, one from each parent. So you have a maternal chromosome and a paternal chromosome. They're the same chromosome carrying the same genes, maybe different forms of that gene or alleles. Okay? These genes are segregated from each other when gametes are formed. So if you go back to the process of meiosis, when you go ahead and produce haploid cells, the sperm or the egg, you will get one from mom or dad, but not both. And number 4, the alleles for different genes usually segregate independently of one another. We will talk a little bit later on about examples where this is not always true, where two genes may be located on the same chromosome, but for basic purposes, when we were talking, especially in Mendelian genetics, we assume that the genes are located on different chromosomes. Now, this is some pretty amazing discoveries that Mendel made, and they were often lost for a while until years later when they're starting to do more research, trying to find some explanation for what Darwin saw in On the Origin of Species by Means of Natural Selection. Today, Mendel's principles form the base on which modern science of genetics has been built. And while we focused on traditional or Mendelian genetics, where you have simple dominance and recessiveness, that isn't always what we see. Okay. So while we were talking last class about just, yes, the dominant covers up the recessive, we don't always get offspring or progeny in the expected Mendelian ratios of 3:1 for heterozygous breeding with heterozygous. And this is because allele expression is not always as simple as dominant alleles overriding recessive ones. So today we're going to go ahead and show you some allele expression that doesn't necessarily follow the basic Mendelian principles. Turns out some alleles are neither dominant nor recessive, and many traits are actually controlled by multiple alleles or multiple genes or even both. So some types of allele expression outside of normal Mendelian inheritance include incomplete dominance. The snapdragons in the upper right-hand corner here are a great example of this. In snapdragons, pink flowers result when you cross red and white. So in particulate inheritance, which is what Mendel was studying, and in many respects he was very lucky to go ahead and identify traits in pea plants that were very simple dominant and recessiveness. In particulate inheritance, the paint colors don't mix. Like if you get one gene and another gene, right, red and white, you don't end up with pink plants. But when it comes to snapdragons, you do. And this is known as incomplete dominance. The dominant allele is not strong enough to overpower a recessive. Likewise, we end up seeing codominance. Now, in codominance, both alleles are going to be dominant, like you see in roan coat color for the cow on the right. And so you see both colors. And in many cases, when we deal with codominance, we also have what are known as multiple alleles. Probably the simplest example to go over for this would be human blood types. A, B, and O. That's 3 alleles for a trait rather than the normal 2 that we covered last class. Then we get to polygenic traits. Now, polygenic traits are those that are controlled by more than one gene. Good examples of these are anything on a spectrum. You think about hair color, skin color, height. You don't just have fixed heights for people. You don't just have black, white, brown. That's it for skin color. There's a whole gradation of intermediates between any of these combinations. Okay, so polygenic traits exhibit a range of options rather than just being turned on or off. And then the last one we'll talk about today is what we call lethal alleles. These are alleles where if you have either the homozygous dominant version or the homozygous recessive version, it simply kills the organism and they're never born. And so we'll take a look at this in terms of populations and inheritance. All right, so let's start out with incomplete dominance. Now, incomplete dominance is neither allele is able to dominate the other, and the heterozygote produces an intermediate phenotype. So between the two homozygous forms. In crosses involving incomplete dominance, the genotype and phenotype ratios are identical. So if you remember when we were looking at pea plant flowers, you had the 3-to-1 ratio, 3 purple, 1 white. Okay? The actual genotype ratio was 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So that 1 to 2 to 1 ratio is what we're going to end up seeing here when we go ahead and look at incomplete dominance. So sweet peas are another example of this, where red and white flowered plants cross to produce pink flowers. So if we start with the flower here on the left, this is going to be homozygous for the white trait. And so you'll see that we don't use the color because there isn't a normal dominant and recessive case here. So we use C for color and then a superscript written up above for the particular color of that particular allele. Okay, so on the left you see CW, homozygous for white. If you go to the far right, we have something very similar. In this case, we have CR, color red, twice. So this is the homozygous for red. If we cross those, so these snapdragons end up producing a pink combination, CR-CW. They have an allele for red and an allele for white, and neither of them is dominant over the other, so it mixes or blends and shows the combination of the colors. Let's go ahead and try this to see if we make sure we understand it. So as I mentioned, snapdragons exhibit incomplete dominance, and red flowers and white flowers are going to be crossed. So if we take a look at that, your left has the red flower, CR, and your right has the white flower. Can you figure out what alleles can possibly be passed on for each one of these parents? Okay, in terms of developing a Punnett square, what options does the red flower have? Good. What options does the white flower have? Good. So hopefully you were able to identify that the red flower can only give CR as an allele in its gametes. Likewise, the white flower can only give CW as an allele in its gametes, which means no matter which sperm, which egg you get, when we go ahead and fertilize the red flower and white flower together, we end up with pink offspring. So what are their allele combinations? Remember, now we're diploid again because we've had fertilization. Yeah, since they're pink, hopefully you recognize we have CR, CW. Okay. Now let's go ahead and try another one. In this particular example, we're going to determine the phenotype and genotype ratios for the offspring from a backcross. And now backcross is where you go ahead and breed an offspring with one of the parents. So we're taking the F1 pink flower and we're going to go ahead and breed it against the red parent. Now, do you remember what the genotype was for the pink flower? Hopefully you were able to come up with CR-CW, and we remember that CR-CR is the genotype for the red flower. Now, when we go ahead and start looking at allele options for the sperm or egg here, if we look at the gametes for the pink flower, we now have options. It's no longer just CR or just CW, but we actually have both CR and CW as options. But the red flower, just like on the previous slide, still only has CR-CR. So take a minute, pause the video, and see if you can go ahead and come up with what the potential offspring are in this case. Use the arrows to help guide your thinking. Hopefully, you were able to come up with 2 red and 2 pink. If you go ahead and follow the lines, the first 2 we can see are CR, CR, and CR, CR. Okay, so we have 2 red, and then we have CR, CW, CR, CW. Okay, so here we go, there we go, and you see that we have 50% red and 50% pink. So that's the basics of how incomplete dominance works. Now let's go ahead and introduce you to codominance. And in many respects, it works the same way as incomplete dominance does, except for how it expresses the phenotype. Okay? But the genotype inheritance portion is the same. So in cases of codominance, both alleles are independently and equally expressed in the heterozygote. Good examples of this include our white and red cow. So just like we saw with the snapdragons, if you have a red bull, it gives you wings. You have gametes that can only be CR. And if you have a white cow, the only gametes that you can have are CW. Pause the video here and think about what offspring this pair is going to have. Hopefully, you are realizing that these are going to end up being a combination, CR, CW. But unlike the snapdragons, we don't see pink cows. Maybe you do. Maybe that's where strawberry milk comes from. No. Okay. What we end up seeing is what's called roan. You see both red and white. And we have the same allele combinations that we saw with the snapdragons, CR, CW. But because it's codominant, they both show instead of blending together like we saw with the snapdragons. So roan cattles are a really good example of this, but we have a similar inheritance when we start looking at human blood groups, A, B, and O. Human blood groups, however, have the additional interesting factor of the fact that they have 3 different alleles, so they exhibit multiple alleles. Okay, let's do a couple more practice with codominance, and then we'll look at some of those other forms of inheritance. All right. So in a cross between 2 heterozygotes, roan short-horned cattle, red, roan, and white offspring are produced in a 1:2:1 ratio. Let's see how that plays out. If you have a roan bull, notice that you have CR-CW as the allele combination, and you have a roan cow, which is also going to produce CR-CW allele combinations. Depending on which one of the gametes you get, sperm from the bull, eggs from the cow, we're going to produce what combination of potential offspring. Pause the video here and see if you can figure it out. Hopefully, you were able to see that we end up with red, roan, and white as options. And they are, as the statement above says, in that 1:2:1 ratio. We have the option of CR-CR, but we have 2 chances to go ahead and end up with CR-CW, and again, 1 option to go ahead and get CW-CW. Now, if you think about this in terms of probability, this would say that you have a 25% chance to get a red cow, 25% chance to get a white cow, and a 50% chance to go ahead and get a roan cow from this breeding combination. All right. Remember, there is a difference between probability and actuality. These are the statistical likelihood, and the more offspring you have, this is the probable outcome you would end up seeing. But if you just have one, that isn't necessarily going to produce in these particular chances. Okay, you could still end up with a white, even though that's a lower likelihood than with a roan. Okay, we'll talk about more examples when we get to class. Okay, let's do another one. So when we deal with codominance, now we're going to go ahead and look at another example of a red bull, this time with a roan cow. All right, so what are the alleles for a red bull? Hopefully you recognize that it's homozygous for the red color, so CR, CR. And what about for the roan cow? Hopefully you recognize that this is CR, CW, it's the hybrid. And we can go ahead and take a look at the potential gametes for each. The red bull can only give out CR, but the roan cow can give you the option of CR or CW. We can follow our arrows, going ahead and combining the two, and what we end up seeing is that we have a 50-50% chance or a 1:1 ratio of either roan to red. But you can't get a white because the red bull does not carry the white allele. So when we start looking at inheritance, We're really looking at the probability of getting a particular allele from a particular parent. Okay, and we can go ahead and identify the diploid allele combinations for these offspring. If it's red, it's going to be CRCR, and if it's roan, it's going to be CRCW. Hopefully this is starting to click and we're starting to get good at making these predictions and understand how the alleles are going to be inherited. Let's go ahead and do another one, this time using a white bull and a roan cow. Ooh, he looks angry. Okay, so what are the alleles for the white bull? Hopefully you said CW, CW. What about for the roan cow? We just did it on the previous slide. Right, CR, CW. And we can go ahead and look at separating out our alleles into the gametes. So the white bull can only produce CW as the allele in its sperm. But the roan cow has the option of producing either a CR or a CW in its eggs. So when we start looking at inheritance patterns for their offspring, we're once again dealing with that 1-to-1 ratio or 50% chance of both of these options, either roan or white. Okay, roan will be CRCW. It got its R from its mom, and the white got CW from both parents. But remember, the dad can only give CW as its color allele to its offspring. Now, many genes have more than just two alleles. We've shown mostly examples with just two alleles, whether it was tall versus short in the height of pea plants, or green versus yellow, or even in these examples, looking at red versus white in cows and snapdragons. This does not mean that you can have more than two alleles, though. Remember, you still only have two sets of chromosomes, one from mom and one from dad, and each set will only have one allele. So even if there are 3, 4, 5 different allele options for a particular gene, any one person can only have two of them. In humans, genes for blood type and eye color are expressed through multiple alleles. And in most cases, multiple alleles are going to exhibit some form of codominance. There may be a recessive from the combination, but then the other alleles will end up typically exhibiting some form of codominance. We also have what are known as polygenic traits. These are those produced by the interaction of several genes. So instead of just one gene with multiple alleles, we're dealing with multiple genes. And when we start looking at this, they are going to show the widest range of options in terms of the phenotype. This is a good example in skin color, in height, in hair color. These are all polygenic traits, which is why there are so many variations. And for skin color, at least, there are at least 5 genes that control this trait. There may be as many as 7 or 8 genes that just control this one trait. The last form of inheritance that I wanted to talk about today is what's known as lethal alleles. Now, lethal alleles are gene mutations that result in a product which not only non-functional, but actually may kill off the organism. Some lethal alleles are fully dominant and therefore lethal in a heterozygote situation as well. Dominant lethal alleles usually become really rare within a population because having them causes the individual to die. We do have an exception, however, in humans. There's a disease known as Huntington's disease, which is a neurodegenerative disorder, and the expression of the allele is delayed until you're an adult. And many people will pass on this trait, not even knowing that they have the disease. So, in other cases, however, the allele mutation results in a viable heterozygote with a recognizable phenotype. So, you end up being able to tell that they have the gene without them having the severe consequence of not surviving. Recessive lethal alleles are only fatal in what we call the homozygote recessive, homozygous recessive, since their effect is masked by the heterozygote and not existent in the homozygous dominant. So let's give you an example of this. When lethal alleles prevent full-term development of the embryo, offspring are produced at a 2-to-1 ratio rather than the 3-to-1 ratio that we have seen before. Okay. In the inheritance of coat color and yellow mice, offspring phenotype ratios don't follow the normal Mendelian 3-to-1 ratio. When yellow mice are mated together, about 2/3 of the offspring are yellow and about 1/3 are non-yellow. So here we see two yellow mice being bred. These are often known as like pinky mice. They're often fed as food to snakes and other animals that eat mice, but they are typically going to be heterozygous for a particular allele. Okay. They have the option to go ahead and produce the dominant Y or the recessive Y, but if the individual gets the dominant, they don't survive. Okay, they're not viable. So the only ones that are born are either the heterozygous or the homozygous recessive. And so this is an example of a lethal allele, but it's not just in mice. We have it in humans as well. And one of the most common ones that we can identify is what's known as brachydactyly. Anybody know what brachy means? Maybe you're familiar with the term brachiocephalic. Um, if you have any dogs, if you have a bulldog or a pug or anything that's got a really short snout to it, they exhibit what is known as brachiocephalic. Their sinus, their nose is actually truncated. It's really short. This is why they have that kind of difficulty breathing. They have that kind of pattern. Okay, so brachy means short. What about dactyly? Have you heard the term pterodactyl? So pterodactyl actually means land digit. If you take a look at a pterodactyl, it's a flying dinosaur, but it looks like it crawls around on the land. And so land digit. So dactyly means digits. So brachydactyly in humans is a shortening of the finger bones caused by a lethal allele. So heterozygotes have shorter fingers than you normally would. But if you're homozygous for this trait, you die in infancy because of all sorts of skeletal defects and don't survive. Okay. Of the offspring of two brachydactylic people, 1 in 4 will die in infancy. 1/2 will show brachydactyly, and only 1 in 4 will be normal. That's our 1 to 2 to 1 ratio that we've been looking at in terms of genotype. So, what does that look like? The picture on the left is an x-ray of a normal hand. You can see how long all the different bones are for the carpals and digits. And then you go ahead and on the right see the individual with brachydactyly, and hopefully you can see from the x-rays that there are a lot of shortened bones here, especially for the middle and later digits, okay, portions of the digits. Now, that's the end of the primary notes, but I did want to kind of get you thinking about how these kind of mutations impact organisms. And if we're going to talk about dactyly, there are several other dactylies we can talk about. One of these is what we call polydactyly. Now, what does that prefix poly mean? That's right. Poly means many. So polydactyly is where you have many digits. Do any of you have a kitten that has an extra finger or toe on the paws? Yeah, it turns out that polydactyly is a dominant trait, and so it's inherited more often. But we see this in animals. We don't, however, tend to see it in humans. Why do you think that is? While you're thinking about it, here are some examples of human polydactylies. Notice that you're dealing with an extra digit, okay, whether that's an extra thumb, an extra toe, or just an extra finger mixed in on the hand. Maybe great for playing piano, I don't know. Not all polydactyly is functional. Some of them are hanging on and are not usable. In other cases, polydactyly can produce usable digits. But when we start looking at this, let's think about how humans don't necessarily follow typical dominance and recessiveness in terms of what's common within the species. Most likely, you've seen a lot of different blue-eyed people. Well, blue eyes turns out to be a recessive trait. Why is it so common if it's recessive, though? It's because people like it. They think it's attractive, and so it gets picked more often than it should. The opposite is true when we start looking at polydactyly. I mean, we don't see individuals walking down the hallway and go, oh, check out that girl. She's got awesome extra toes or anything like that when she wears sandals. And because of this, it tends to not get passed on, not because it's not dominant, because it's less likely to get selected due to some preference within the individuals of the population. We can also start looking at webbed digits, for example. This is a good example of this is that you can start seeing that this is a form of what we call syndactyly. This is actually related to something that we call ectodactyly. Um, there was famously a tribe up in Zimbabwe in Africa that were known as the ostrich people, kind of a derogatory term, but they had an extreme case of ecterodactyly. And because their gene pool was so small, because they were so remote as hunters and gatherers, they ended up passing it on, and it was like a quarter or maybe even higher, 25% or higher of the population actually had these type of feet. And when explorers actually came across their footprints, they were thought they were either giant ostriches or some sort of alien species. And there's interesting stories about the history of these people and how they feel they got here, and how other people feel like they got here as well. So in the optional activities, there is more research you can go ahead and do on these people from Zimbabwe, known as the ostrich people. But think about it in terms of genetics, of inheritance, and then of selection, and especially with humans, the impact human perception of beauty or attractiveness impacts the selection of desired traits. Okay, that's all I got for you guys today. Hope you have a great rest of your day. Take care of yourselves. Stay healthy. Bye.